Hello, everyone. Welcome to Global IMMUNOTALK 2024. I'm Kazuyo Mara from Graduate School of Medicine, Osaka University, Japan. So today, we're happy to have Yumi Kim from Korea. But before moving to her talk, I have two announcements. One is about how to make a question. So in this global talk, we unfortunately cannot directly take a question. So if you have any question to Dr. Kim, please go through the Twitter. I'm going to explain how to do it after her talk. And another information is about next week's speaker. Next week, we will have Padmani Sharma from the University of Texas. So please don't miss it. So now I'm so honored and happy to introduce Dr. Yumi Kim. So Dr. Yumi Kim is a associate professor at KAIST, Graduate School of Medical Science and Engineering in Korea. She earned both her bachelor's and master's degree in pharmacy from Seoul University in Korea, and then obtained her PhD from Thomas Jefferson University in US. Before joining KAIST in Korea, she held postdoctoral position at Harvard Medical School and Whitehead Institute, followed by the faculty position in the PostTech in Korea. So her research has significantly advanced our understanding of signaling and molecular biology, particularly in the regulation of nucleotide sensing toll-like receptor through intercellular trafficking and the localization. So now I'm happy to call Dr. Kim. Yumi, could you please turn on your camera? Okay, perfect. And so thank you very much for coming to Global IMMUNOTALK.
Thank you, Kazuyo.
Okay. And so before moving to your talk, we usually make one question for the speaker to let all the people know what kind of like a thought you have. So today I have one question for you. And the question for you is, what do you believe the most critical skill for the scientist to develop their early career? So could you answer to this question?
Okay. Well, I guess there are so many skills you need to have to build a successful career as a scientist. But one thing I would like to emphasize today is to have strong curiosity. I guess I believe that the fundamental foundation of science is really curiosity. First of all, without it, you cannot form good questions. More practically, I think curiosity can lead you to unexpected discoveries and often give you the answers why your experiment did not work. So as an experimental scientist, we usually form a hypothesis and try to prove it by doing experiments. But the experiment results can be often puzzling. And I think those unsuccessful experiments can be turned into a new discovery by curiosity-driven thinking and open mind. And also, I think without your own curiosity, research can become very onerous and uninteresting. So I would like to emphasize curiosity, building
So
curiosity.
how can the student can get the good curiosity? How can we educate to let
I
them?
know it's not easy. I mean, I think actually the students who decided to become a scientist usually already have a pretty good curiosity-driven thinking. But I tell my students, try to ask questions during seminar or try to form questions while reading papers. I think they forced to think more about the questions. And I think curiosity is a kind of skill you can also build in doing so. Or probably there are many other ways as well.
Okay, wonderful comment. Thank you very much. Okay, so let's move to your talk. Would you mind sharing your slide? Please
Okay.
take a time.
Okay.
Yeah, perfect.
Can you see it?
Okay.
Okay, good,
Yep.
great.
Okay.
Okay, so before I start, I would like to thank Kazuo again, and I also would like to thank other organizers of the Global Immunotalks for inviting me to give a talk. So my lab is mostly interested in elucidating the molecular mechanisms underlying the innate immune regulation, and recently we became interested in eosinophils. So today, I will talk about our recent findings on GPR43-mediated regulation of eosinophils. Okay. So as you know, eosinophils are classified as granulocytes because they have a lot of granules. And these granules are called specific granules or secondary granules and have high levels of cationic proteins which binds to acidic dye called eosin, and the name comes eosinophil. And these cationic proteins include major basic protein, MBP, and eosinophil specific peroxidase, EPX. And these proteins are often used as eosinophil specific markers. Additionally, various cytokines, chemokines, and growth factors are stored in these granules. And upon activation of eosinophils, they become rapidly secreted. Eosinophils also produce various pro and anti-inflammatory lipid mediators. And of course, eosinophils also express various pattern recognition receptors, chemokine receptors, Fc receptors, complement receptors, and adhesion molecules like any other myeloid cells. Historically, eosinophils are best known effector cells against parasite infection. Upon activation by complements or IgE bound to parasites, eosinophils secrete granular contents and they damage parasite cell membrane and kill them to protect the host. On the other hand, eosinophil activation can play pathogenic roles in case of various allergic diseases. For example, during allergy airway inflammation, IL-5 secreted by ILC2 or Th2 cells recruits eosinophils into the lung. And effector molecules from eosinophils promote epithelial damage, goblet cell hyperplasia and mucus secretion, and tissue fibrosis. So therefore, in both parasite infection conditions and allergic diseases, eosinophils are considered as pro-inflammatory cells that are recruited to the tissues only after the initiation of inflammation. However, more recent studies show that eosinophils are found in multiple organs, even at steady state. Eosinophil development occurs in bone marrow, and differentiation of eosinophil precursor cells and mature eosinophils absolutely depend on transcription factor called GATA1. And the mature eosinophil express a surface protein called SIGLAC-F, which is often used as a eosinophil marker. Eosinophils also express IL-5 receptor and CCL3, which is a receptor for eotaxins. And IL-5 secreted by ILC2 or eotaxins secreted by epithelial cells and stromal cells recruit eosinophils in tissues. And in each tissue, eosinophils are estimated to live up to a week and play tissue-specific homeostatic roles. So for example, in 2011, Richard Roxley group reported that in the steady state white adipose tissue, eosinophils are accounted for 90% of IL-4 producing cells. And by promoting IL-4 dependent M2 macrophage polarization, eosinophils play essential roles in maintaining glucose tolerance and body's metabolic homeostasis. Indeed, the number of eosinophils in adipose tissue were inversely correlated with mouse body weight. And upon high-fat diet, mice that do not have eosinophils due to mutation in GATA1 gene gain much more weight than control mice and could not handle glucose properly. Subsequently, other studies have also shown that eosinophils have various organ-specific roles, such as apoptotic cell clearance in the thymus, mammary gland development, CD4 T cell regulation in gastrointestinal tract, and maintenance of plasma cells in the bone marrow. However, we have limited understanding how these various functions of eosinophils are regulated. So while thinking about the regulatory mechanism of eosinophils, we came upon with a specific receptor called GPR43. So GPR43 is a receptor for short-chain fatty acids such as acetate, propionate, and butyrate. It is also called free fatty receptor 2, FFR2, and it is a GI and GQ coupled receptor. Interestingly, GPR43 is very highly expressed in eosinophils and neutrophils. For example, in this example, the reanalysis of public single-cell RNA-seq data of esophagus from eosinophilic esophagitis patients showed the highest expression of GPR43 in eosinophils and neutrophils. As you know, short-chain fatty acids are produced by fermentation of dietary fibers by gum commensal bacteria. They affect host physiology by activating specific receptors, including GPR43, and by inhibiting HDAC. In case of acetate and butyrate, they can also be converted to acetyl-CoA and directly be used as energy source by entering TCA cycle. And many studies show that this short-chain fatty acid can promote epithelial barrier function, induce Treg and tolerogenic dendritic cells, and inhibit inflammatory cytokine production. So therefore, short-chain fatty acids are thought to be mainly anti-inflammatory in general and protect host from various inflammatory diseases. So indeed, epidemiologic studies show that low acetate level in infant is associated with significantly higher risk of atopy disease and asthma. And in mouse studies, low fiber diet aggravate the allergic airway inflammation, while high fiber diet reduces pathology and symptoms of the disease. And similarly, administration of high fiber diet or short-chain fatty acid directly induce the oral tolerance and protect the mice against food allergy. So among the target of the short-chain fatty acids, GPR43 is thought to be one of the most important targets. The Charles McKay group generated GPR43 whole body knockout mice and reported that these mice show enhanced inflammatory diseases, including ova-induced asthma model. However, in these studies, which cell type mediate anti-inflammatory function of GPR43 was not identified. So we analyzed the public data and found that GPR43 expression becomes even more unregulated in eosinophils in asthmatic condition. Also, human eosinophil express almost exclusively FFR2 encoding GPR43 among different free fatty acid receptors. So therefore, we hypothesized that GPR43 may play a critical role in regulating functions of eosinophils. So to study the role of GPR43 in eosinophils, we generated eosinophil-specific GPR43 knockout mice by crossing the fluxed GPR43 mice with EPX-CRE mice. And by using ROSA UIFP mice, we found that the YFP expression is specific in eosinophil. In other words, the CRE deletion only happens in the eosinophils. And also by qPCR analysis, we found that eosinophil-specific GPR43 knockout mice generated the eosinophil has no GPR43, similar to the GPR43 whole body knockout eosinophils. And we also tested the functional assay using calcium flux assay. So wild type eosinophil induced the calcium flux upon stimulation with acetate, and this calcium influx is completely gone in GPR43 deficient eosinophils. So in these mice, we first look at the lung immune cells at steady state. And as you can see, at the steady state, we didn't not see any major functional differences or cell differences. So eosinophil numbers or Ig producing B cell number, ILC2 numbers are all same as the wild type cells. And also CD4 T cell population was not disturbed in this eosinophil specific GPR43 knockout mice. So next, we challenged mice with house dust mite to induce the asthma. Interesting, in these cases, we found that the immune cell infiltration into the lung was highly increased. And not only eosinophils, but also we found the neutrophil infiltration was increased. And lung pathology shows that the increased mucus production and high fibrosis. And as expected from these results, the airway resistance by a metacolin test showed that the airway resistance was more aggravated in this GPR43 specific, eosinophil specific GPR43 knockout. Asthma can be divided into roughly several endotypes, and most of asthma, especially mild asthma, show eosinophilic endotype. In these examples, usually allergen stimulation of epithelial cell induced production of alarmins such as IL-33, IL-25, or TSLP. And these cytokines activate ILC2 or Th2 cells. And again, the type 2 cytokines from these cells recruit and activate the eosinophils, and then leads to eosinophil induced damage of epithelial cells and other symptoms. And these eosinophilic asthma is usually can be treated by steroid administration. However, other types of asthma, for example, the asthma induced by microbial infection or pollutants, smoking, and obesity-associated asthma, instead of type 2 cell activation, we usually see Th1 or Th17 cell activation. And IL-17 from Th17 cell can recruit neutrophils, and neutrophil is now major effector cells in this neutrophilic asthma. One feature of this neutrophilic asthma is they are not treated by steroid. And also more severe asthma case, they show the mixed granulocytic asthma, and they have a lower lung functions. And again, these mixed granulocytic asthma are resistant to steroid. So we thought that it was interesting that in our eosinophil specific GPR43 knockout mice, we saw not only eosinophil increase, but neutrophil increase. And when we look at the CD4 T cells, we saw increase of GATA3 positive CD4 T cells as well as aura gamma T positive CD4 T cells. And cytokine analysis showed that increase in IL-4, 5, 13, as well as IL-17. So in these knockout mice, we see both Th2 and Th17 activation. So in order to look at the immune cell landscape change in more unbiased way, we performed a single cell analysis of immune cells in the lung after asthma induction in both wild type and eosinophil specific GPR43 knockout mice. Unfortunately, the eosinophils cannot be seen in these single cell RNA sequencing data, and this is probably due to very high RNA content in eosinophils. And most of the single cell data in mice as well as in human, it's very difficult to see eosinophils. So nonetheless, we were able to detect all other immune cell subsets here. And among these immune cell subsets, the biggest difference we can detect was neutrophils. And when we cluster the neutrophils alone, we could see that neutrophil can be divided into two clusters, cluster 0 and 1. And as you can see, in this eosinophil specific GPR43 knockout lung, not only the number of neutrophils increased, but also we could see this increasement in the cluster 1 type of neutrophils. And among various genes, we found that these neutrophils highly expressed the PCAM1 as well as Siglec F. So I told you that Siglec F is usually used as a marker for eosinophil. But interestingly, in this example, these neutrophils also highly express the Siglec F. And not only Siglec F and PKM1, these cluster 1 neutrophils also express gene for TIM3 and other genes. And instead, they kind of downregulated the classical neutrophil-associated genes such as retinol G or MMP8. And so from now on, I will call this neutrophil subset as a Siglec F neutrophils because the Siglec F positive neutrophil has been already described. And as early as 2017, the people found that neutrophils expressing Siglec F in lung cancer. So in these KP tumor lung adenocarcinoma model, they found that some neutrophils highly express Siglec F. And when they did the transfer experiment using conventional neutrophil versus the Siglec F high neutrophils, they found that Siglec F high neutrophils promote higher tumor growth. And also in neutrophilic asthma induced by the diesel exhaust particles, the neutrophil were shown to express Siglec F highly. And in this paper, they showed that the Siglec F high neutrophil has more pathogenic features such as NET-associated pathway genes were highly unregulated. And in human allergic asthma patients, the more severe symptom-containing patients showed higher level of Siglec F high neutrophils. So in general, Siglec F high neutrophils are recognized as a more pathogenic forms of neutrophils compared to conventional neutrophils. And indeed, when we compare the conventional neutrophils versus Siglec F high neutrophils in our experiments, we could only also see that the Siglec F high neutrophils highly express NET-associated pathway genes, angiogenesis genes, and myeloid cell recruitment associated genes, as well as extracellular matrix remodeling associated genes. So we confirmed that indeed these Siglec F high neutrophils are present by doing FACS analysis. And as you can see, Siglec F positive or PKM1 positive or double positive neutrophils were highly increased in the lung of asthma-induced mice in case of deletion of GPR43 in eosinophil. Also, when we compare lung versus valve, these neutrophils were more highly found in the valve fluid. Interestingly, when we compare the lung versus blood, we could not see any of these Siglec F high neutrophils in the blood. Therefore, we believe that the differentiation of Siglec F high neutrophils occurs in the lung in situ. So, of course, our question is how these GPR43 knockout eosinophils induce emergence of these pathogenic Siglec F high neutrophils. So we sorted the eosinophils from both wild type and eosinophil-specific GPR43 knockout mice after asthma induction. And as you can see, these GPR43 deficient eosinophils show many vacuoles, and these vacuoles are indicative of degranulation. And also, when you stain the nucleus using propionate iodide, you could see that very disorganized nucleus morphology. And these are the typical features of hyperactivated eosinophils. So by just looking at the morphology, we could tell that the lack of GPR43 induces the hyperactivation of eosinophils. And we also performed the bulk RNA sequencing of the sorted eosinophils and could find that the genes associated with cellular metabolism, hypoxia, and many genes associated with inflammation were highly unregulated in knockout eosinophils. So next, our question was whether the eosinophil can directly induce differentiation of this conventional neutrophil into the Siglec high neutrophils. So we performed the in vitro co-culture assay using neutrophils sorted from the bone marrow with eosinophils from asthmatic lung. And as you can see, wild type eosinophil could induce CGLF high neutrophil differentiation. However, when we used the GPR43 knockout eosinophil, the efficiency was much more higher. So we could show that eosinophil has direct effect on neutrophils. So next, we wanted to see what are the mediators of the CGLF high neutrophil differentiation. So again, using in vitro system, we stimulated the conventional neutrophils using different cytokines. And we could find that incubation with IL-4 can induce PKM1 expression. In contrast, incubation with IL-4 did not induce CGLF. Instead, CGLF expression required GM-CSF. And when we co-added GM-CSF as IL-4, we could see much higher expression of both PKM1 and CGLF. And again, using in vitro co-culture assay of neutrophils with asthmatic lung eosinophils, we found that when we block the IL-4 and GM-CSF by using neutralizing antibody, we could significantly inhibit the conversion of conventional neutrophil into this CGLF high and PKM1 high neutrophils. So we believe that these two cytokines are major soluble mediators of eosinophils. And the next, we also look at the cytokine expression in other cytokine expression in these lung eosinophils. And we found that actually among the top 60 asthmatic lung eosinophil cytokines, many of them belong to the chemoattractants for neutrophils. And as you can see, when neutrophils were co-cultured with eosinophils, they form these clumps. And even in vivo, we could see that the eosinophils make a direct contact with the neutrophils. And here I show you some examples of neutrophil chemoattractants by qPCR analysis, or I'm sorry, from the single cell analysis. And as you can see, compared to wild type eosinophils, these GPR43 knockout eosinophils express these neutrophil chemoattractants more highly. And again, the co-culture assay showed that compared to wild type eosinophils, this GPR43 knockout eosinophils induced more aggregation of neutrophils. And finally, we did this transwell migration assay by having neutrophils at the top well and the eosinophils at the bottom well. And the neutrophils were stained with the CTB. And here I show you the fluorescence images. So these are only the neutrophils. And as you can see that when you have the eosinophils that lacking GPR43 at the bottom well, that the neutrophil migration was much more enhanced. So in other words, this GPR43 deficient eosinophil can recruit neutrophils very actively. And finally, we tested whether the neutrophil and eosinophil direct contact is required for conversion of conventional neutrophil into the CGLF high or the PKM1 high neutrophils. And as you can see, when we co-culture the neutrophils and eosinophils in separate well, in this case, we used a smaller trans well so that the neutrophil cannot migrate through the pores. And in this example, we see completely blocked conversion of or the differentiation of CGLF high neutrophils. So with this, I show you that eosinophil can induce differentiation of CGLF high neutrophils via IL-4 and GM-CSF and contact-dependent manner. And these events happens much more highly using GPR43 knockout eosinophils. So finally, we tested the function of this signal of high neutrophils. And the reason we decided to look at these is because when we look at the cytokine expression in total immune cell population, we found that actually the expression of IL-1 alpha and TNF, and especially IL-23A, is most significant in neutrophil, and especially these CLF high neutrophils. And also these neutrophils highly express TJ beta gene as well, and IL-1 beta gene. Because TJ beta, IL-1 beta, and IL-23, these all are associated with differentiation of pathogenic Th17 cells, we asked the question whether there is any correlation between the CLF high neutrophils with Th17 cells. And indeed, we found that in the lung of asthmatic mice, number of or the proportion of CLF high neutrophil were highly correlated with proportion of IL-17A producing Th17 cells. So we tested whether the neutrophil can directly induce Th17 differentiation. So for this, we first purify bone marrow neutrophils and then let them sit for 24 hours by themselves, or we stimulate them with IL-4 and GNCSF so we can produce CLF high neutrophils. And then we co-culture these neutrophils with naive CD4 T cells in the presence of TJ beta and IL-6. And as you can see, when we have wild type or conventional neutrophils, we do not induce increasement of Th17 cell differentiation. However, the CLF high neutrophil can unregulate the differentiation of Th17 cells. So we believe that among many other functions of the CLF high neutrophils, we believe that promoting Th17 differentiation is one of the important function of the CLF high neutrophils in asthmatic lung. So in summary, I showed you that the eosinophils that lack the GPR43 could convert the conventional neutrophils that probably come into the lung during this asthmatic airway inflammation. And these conventional neutrophils differentiated into or converted into CLF high neutrophils via IL-4 GMCS dependent as well as contact dependent manner by these eosinophils. And IL-1 and 23 produced by the CLF high neutrophil can produce Th17 cells. And as well known, these Th17 cells then turn back to neutrophils and recruit these neutrophils or activate these neutrophils. So there is a vicious cycle going on. And also, although with the time limit, I do not show any data regarding the increasement of Th2 or the molecular mechanism behind the Th2. We have data that the knockout GPR43 knockout eosinophils can also promote the Th2 responses. So this specific depletion of GPR43 in eosinophils can result in increase in both Th2, Th17 cells, as well as more recruitment of neutrophil and conversion of neutrophils into these more pathogenic CLF high neutrophils. And these cells together probably promote more severe lung inflammation. And so I can summarize this as GPR40 signaling in lung eosinophils are usually very critical for suppressing hyperactivation of eosinophils and prevention of neutrophilic activation. And we are thinking that perhaps activating GPR43 in eosinophil or in general can be maybe a good therapeutic strategy, especially in neutrophilic asthma. So with this, I'd like to end my presentation by acknowledging the people who did this work. And actually, my talented graduate student, Jihyun Yu, almost single-handedly performed all of these experiments with the help of Song Young Kim, who helped the single-cell analysis. And these are my students. And I also would like to thank John Park at KAIST. He helped our single-cell analysis. And Haeyoung Kim at Seoul National University, she gave me a lot of guidance on eosinophil study as well as asthma induction, because this is my first adventure into study of eosinophils and asthma and allergic diseases. And this EPX Cree mice was kindly given to us by the late James Lee at Mayo Clinic, and this is my funding source. Okay, thank you very much for your attention. Oh, Kazuo, I think your microphone is off.
Sorry, thank you very much, Yumi. It was a really nice talk, and I personally have a lot of questions to you, but unfortunately, we can't do that. So you can turn off your camera. Thank you very much again. I would like to end with sharing one slide about how to make a question here. So if you have any question want to ask to Dr. Kim, please go through X. I told you it's Twitter, but now it changed to X. So through the X, and first search for the account Global Immune Talks, and then find tweet that says ask question for Dr. Kim here. And then please reply to that Twitter what you want to ask. And please mention Global Immune with that. Okay, so that's all. And thank you for joining. See you next week.